A very good day. My name is Anishifa Atira binti Ahmad Fazil and my supervisor name is Dr. Ngiang Singh. So today I'm going to present my research study entitled T residue for Lacase enzyme production by Marasmus Cardophilus Limas MSA. This is my abstract for my research study. So lacase enzyme is widely used enzyme, therefore it is highly demanded. So many researchers are trying to find alternative where they use lignocellulosic waste as a substrate in the fermentation process to produce lacase. However, not many have looked into the effect of spent tea residue as substrate in fermentation process. So the aim of this study is to investigate the suitability residue as co-substrate in both solid state and submerged fermentation for the production of lacase by Marasmus Cardophilus Limas MSA. From the result obtained, SSF was found to give rise to the highest level at lacase enzyme activity, which reaching 0.001 to 195 BD per microliter on day 15. It showed that T residue can be an interesting substrate for the production of lacase enzyme by M. cardophilus. So next, we move on to the introduction part. Here is my research background of my research study. It's about Marasmus cardophilus, which is a species of fungus in the family of Marasmaceae. It is known as white rot fungi called Basidiomycete, which has the most abundant of lacase enzyme. According to Wang et al. 2019, lignocellulose waste is a good substrate because it can be a nutrient source, a support, and also an inducer in producing lacase using solid state fermentation. So an example of lignocellulose waste is tea residue. Tea residue is the tea after being used for making drink or deprocessing. There are two types of fermentation which can be used in producing lacase enzyme by fungus, which are solid state and submerged fermentation. Solid state fermentation is a process where it can occur in the absence of water or near absence of water, while for submerged fermentation is a process involving cultivation of microorganisms in the liquid proof. Next slide is my problem statement. As we know, lacase enzyme is multifunctional enzyme. Therefore, it is needed in a huge quantity. So when the demand is high, the cost of lacase production needs to be reduced. Therefore, many different kinds of agricultural waste have been tested, and some lignocellulose waste have been reported to enhance the production of lacase. But there is little that is known about the feasibility of using tea residue as substrate in producing lacase. So we have two objectives, which the first one is to investigate the effect of tea residue on lacase enzyme production, and the second one is to investigate the effect of fermentation technique, which are SSF and SMF. The hypothesis of my research study is tea residue is a good option as a substrate to enhance the production of lacase enzyme by M. cardophilus Limas MS8. Next is literature review. This part I'm going to simplify it. Lacase enzyme is first discovered by Yoshida in Japanese laboratory called Ras Benicifera. A lot of research papers have discovered the advantage of using lignocellulosic waste as an enhancer of lacase production. One of the most abundant of lignocellulosic waste is tea residue. The most abundant compound in tea leaf is polyphenol, which have about 30 to 40% of its dry weight. There are other important components which are lignin, protein, cellulose, and hemicellulose. Based on research paper by Zui et al. 2020, his team has conducted a research using tea residue as substrate and support for the production of lacase by Thermex versicolor. They used solid state fermentation for the mode of fermentation. From the experiment, they successfully proved that tea residue have a potential in enhancing the lacase production. SSF and SMF are the methods that are commonly used. There are many research papers that use both of these fermentation for producing lacase enzyme, and it shows different kind of results. Next is materials and methods for my research study. First is microorganism. In this research study, Marasmus cardophilus Limas MS8 was used. Second is the preparation of raw material. The tea residue was washed with plain water to remove any debris and dry in the oven for 1 hour 10 minutes at 150 degrees Celsius. There is inoculum preparation where the fungal inoculum for fermentation was prepared by growing in the MEA plate until it reached full bleed. Fourth is fermentation, where both fermentation were used. For solid state fermentation, the substrate was moistened 70%, while for the submerged fermentation, 100 of the medium Pasango Australia was added. Both fermentation were incubated static at room temperature for 15 days and sampling for every 3 days. Fifth is sample analysis. For submerged fermentation, 3 ml was taken from each bottle, while for SSF, 0.3 gram was taken out and resuspended in medium of Sangal Australia. And for the last method is lacase activity assay. This part was carried out based on the oxidation of glycol, and the R value was recorded using RGB analyzer called colorimeter. Then calculate the absorbance using Formula 1. After absorbance was obtained, the lacase enzyme activity was calculated using Formula 2. The figure 1 show that the Holmium spectrophotometer set up using phone RGB analyzer. Here is the important part: results and discussion. So Marasmius caudiphyllus humus MS8 has been cultured on mold strain agar, as shown in the figure 2. As can be seen, the fungus grew well for 10 days, and the appearance of single growth morphology and pattern show that organism is pure and free of any form of fungal contamination. So next is the lacase enzyme activity for submerged fermentation. From my observation, the graph as shown in the figure 3, it shows that the lacase enzyme activity is decreasing for a sample and there is no activity for the control. And the decreasing of lacase activity is because of there is probability of contamination, whereas can be seen in the figure 4, the top layer of solution appears in green color, which it is supposed to be in white color. So the probability of contamination might affect the growth of Marasmius caudiphyllus, and it led to the poor production of lacase enzyme. Next is the factor that affect the lacase enzyme activity is cross-contamination. Cross-contamination is defined as the transfer of microorganism, either direct or indirect, from one surface to another. Next, we move on to the lacase enzyme activity for SSF. So here is the result obtained after 15 days of incubation, as can be seen in the graph. Lacase enzyme activity was increased for sample and control. However, the lacase activity for control was not as high as the one with the T residue, which is the sample. From this observation, it indicates that T residue, which act as substrate, is enhancing the lacase production by the fungus. From the figure, we can see the formation of white color mycelium mat, which means that the fungus are well grown on the substrate. Here are some factors that contribute to the increasing of lacase enzyme activity. The first one is lignin degradation. According to Wang et al. 2015, the lignin can limit the fermentation product. There are similar experiment that was conducted by Zhu et al. 2020, where they used T residue as substrate for fermentation process. From the experiment, they observed the degradation of lignin, where the component of lignin is 13.6% before the fermentation process, and after 7 days, it has been degraded, resulting 8.3% of lignin. The fermentation product show an increasing with the degradation of lignin. This indicated that the presence of lignin can trigger the lacase enzyme production, because the fungus will tend to secrete lacase enzyme to degrade the lignin component of the plant cell wall. The second factor is temperature. Based on a research paper by Risdianto et al. 2012, it stated that increase of incubation temperature led to the increase of lacase activity, but it will decrease when it reaches the maximum temperature. In the research paper, they used Marasmius species, and it showed that it can produce lacase at room temperature. In this experiment, the incubation temperature is at room temperature, and from the result, it showed that the lacase activity is increasing, which indicate that the room temperature is the suitable temperature to produce lacase and also for the growth of the fungi and caudiphyllus. The last factor that might contribute to the increase of lacase enzyme activity is environmental condition of fermentation. According to Yazzie et al. 2017, fungi and yeast are the most used microorganisms in solid state fermentation due to their ability to live in the low moisture environment. In this study for solid state fermentation, the substrate was moistened to 70% using the medium selected by Stonga Ashvili. This indicated that the moisture content is suitable for the growth of the fungi and caudiphyllus, and it led to the increasing of leachate and sensitivity. And finally, my conclusion part is, from the research study, we can conclude that SSF gave the highest leachate production compared with SMF. Therefore, the utilization of the residue is a good alternative substrate, which could enhance the leachate production in fermentation process. So here is the list of references that I use in the presentation. And last but not least is, I want to say thank you for my supervisor for helping me throughout this final project. So thank you for watching, and bye.